Hi, my name is Iulia Tapescu, and I am an MD PhD student at Penn on the neurosurgery radiation. We all know about the use of mRNA vaccines for COVID, but what you might not know is that the technology behind mRNA vaccines was pioneered by Professor Kariko in the neurosurgery of Himelac with Professor Wiseman and partners in the industry. Originally, the mRNA vaccine platform was developed for cancers. So now that the COVID pandemic is under control, the question is whether we can harness this technology for cancer therapies. So what we'll do today is tell you where we are with applying the mRNA vaccine platform for brain cancers. Anti-cancer vaccines could be divided into four main subtypes: cell, viral, peptide, and nucleic acid-based vaccines. And today I'll focus on mRNA vaccines. RNA vaccines are made of mRNA molecules that encode tumor antigens. The vaccine is injected in the body and taken up by antigen-presenting cells. The mRNA is translated into protein antigens and then processed into antigen peptides, which associate with MHC class I molecule in the endoplasmic reticulum, and are transferred to the APC surface, thus activating CD8 T cells. In addition, protein antigens, which are sorted through the endosomal pathway, can activate CD4 T cells via the MHC class II presentation pathway. Finally, the secreted protein antigens encoded by the mRNA vaccines can stimulate B cells to produce neutralizing antibodies. Therefore, mRNA vaccines have the potential to create robust humoral and cell-mediated response against tumors. There are four different aspects to RNA-based vaccines that refer to tumor antigens, vector design, delivery, and manufacturing. In this talk, I'll primarily tackle antigens as a way to specifically apply mRNA vaccines for brain tumors, but I'll briefly discuss the other aspects of a mRNA vaccine. New antigens refer to antigens that are completely unique to the cancer cells. Therefore, they're seen as unique by the immune system. In particular, T cells primed to recognize these new antigens attack and destroy cancer cells expressing these mutations. In terms of how the RNA vector is designed, there are a few different strategies. The conventional mRNA encodes the vaccine immunogen and flanking 5' and 3' UTRs, in addition to a 5' cap and poly A tail that are optimized for maximum stability and translational potential. An antigen is translated from the non-replicating transcript. Another strategy involves self-replicating RNAs, which encode 5' and 3' CSC elements, PNSP1 and 4, and subgenomic promoter of an alpha virus, and the vaccine immunogen. Following in situ translation, in addition to the antigen, an RNA-dependent RNA polymerase is also translated, which amplifies the vaccine encoding transcripts, leading to accumulation of tumor antigens within the cell. Transamplifying mRNAs use two different transcripts, one encoding the RNA-dependent RNA polymerase, and the other one encoding the CSC and the vaccine immunogen, thus achieving a similar self-amplifying vaccine. There are different delivery systems for RNA viruses: lipid, polymer, and emulsion-based delivery systems that use cationic molecules to deliver the anionic RNA across the membrane. The LNP system is the most potent one used in vaccines, and they utilize a pH-sensitive cationic lipid system. And together with the RNA, they're taken up in cells through receptor-mediated endocytosis. The potential therapeutic role of RNA vaccines in a solid tumor has been recently shown against pancreatic cancers. Pancreatic cancer is the fourth leading cause of death in the US, with only 30% of patients having a 5-year survival after resection of the tumor. However, only 15 to 20% of patients are actually eligible for surgery. And of note, pancreatic cancers are generally thought not to respond to immunotherapy. In this phase 1 clinical trial, the pancreatic tumor was resected, sequenced, and at least 5 new antigens were identified, and a personalized vaccine was produced for each patient using a conventional mRNA strategy. All patients also received a dose of an immune checkpoint inhibitor, And at 9 weeks after surgery, the patients were given their personalized mRNA vaccine. T cells that recognize specific neoantigens from the mRNA vaccines were detected in the blood of half of the people in the trial. These individuals were termed immune responders. And remarkably, in all the immune responders, there was no evidence of cancer recurrence at a median follow-up time of 18 months after surgery, compared to a median of 13.4 months of recurrence in non-responders. The study emphasized the incredible potential of mRNA vaccines, potentially in combination with immunotherapies for difficult to treat solid cancers. Another new role for mRNA-based anti-cancer therapies has been shown in combination with CAR T cell therapy. The Epstein group at Penn has really shown that CAR T cells can be produced in vivo by using an mRNA-based technology. This mRNA delivery is more efficient. They can be produced quicker and less expensive. In addition, the traditional mRNA vector is expressed more transiently, which is particularly important for the brain where CAR T cells can be particularly toxic. Taking advantage of this technology, Foster et al. At CHOP identified GPC2 as highly expressed in multiple pediatric brain cancers and generated GPC2 targeting CAR T cells using mRNA. Compared to the CD19 control, mRNA CAR T targeting GPC2 was associated with significantly prolonged survival in mice without toxicity. But what about other more aggressive brain tumors? So glioblastoma is a fast-growing solid tumor derived from glial cells in the brain. Based on the WHO classification, is considered high-grade glioma grade 4. It is the most common brain tumor, and it has the worst prognosis with a 5-year survival of 6.9%. The therapeutic standard of care involves maximal safe resection, a fractionated radiotherapy, and chemotherapy. And similar to pancreatic cancer, glioblastoma is generally considered not to respond to immunotherapy. One of the biggest challenges in developing immuno-based therapies against GBM is the immune suppression found in many patients. GBM patients are systemically immunosuppressed. These patients have very few T cells in circulation, have small spleens, and the remaining T cells lack responsiveness against neoantigens. In addition, GBM tumors can cause local immunosuppression by releasing immunosuppressive cytokines, including TGF-beta, prostaglandin 2, and other immunosuppressive cytokines, and have high PD-L1 expression. In addition, steroids and chemotherapy used for standard therapy for other immunosuppressed patients. A further challenge in glioblastoma is related to tumor antigens. Glioblastoma is associated with a wide range of biomarkers, but overall, it lacks a defined mutation for targeted therapy. In addition, glioblastoma lacks extensive genetic instability of high mutation burden tumors, so it does not present with a wide range of immunogenic neoantigens. However, several recent papers have sought to expand the number of neoantigens that can be targeted using mRNA vaccines. In a study from 2022, Zhu et al. Used RNA-seq data from glioma patients from the Cancer Genome Atlas database and identified over 900 potential tumor antigens that were mutated and upregulated. Out of those, 17 were significantly correlated with overall survival, and 14 were also associated with the progression-free survival. Subsequently, the association between gene expression and infiltration of gene-presenting antigens were analyzed, and 10 candidate tumor antigens were finally selected. The authors further studied the immune landscape of glioma patients and found different immune subtypes, or IS, based on molecular signature. IS4 and 5 had the poor survival and correlated with high mutation burden, and IS1 and IS2 corresponded to low-grade gliomas. And given their immune signature, patients with these subtypes were considered to be most likely to respond to mRNA vaccine. In another paper, Lin et al. Used data from the Cancer Genome Atlas and the Chinese glioma genome atlas and identify over 1,000 genes that overexpress, amplified, and mutated in GBM. The authors narrowed down to 56 genes associated with overall survivor, and 6 of those genes were higher in GBM patients, and their levels correlated with the abundance of immune cells, suggesting they could be good vaccine candidates. The authors also identified 3 immune subtypes based on distinct cellular, molecular, and mutational burden. The IS3 subtype was immunologically cold and associated with the worst prognosis, but had high PD-L1 expression, suggesting that these patients could benefit from immune checkpoint therapy before vaccination. Another study by Wood all is GBM data from patients from the Cancer Genome Atlas and the Rembrandt microarray. And these authors identified 9 potential tumor antigens associated with poor prognosis and infiltration of antigen-presenting cells. Similarly, the authors identify 4 immune subtypes ranging from hot and cold. Interestingly, the authors also show that treatment with immune checkpoint inhibitor can lead to switching from a cold to a hot phenotype, which is more likely to respond to mRNA therapy, suggesting that combination of immune checkpoint inhibition and mRNA vaccine may be successful in overcoming the GBM immunosuppression. So there are many ongoing trials for cell therapy, tumor vaccines, or combined therapies in GBM. And with the recent studies I presented on neoantigens, additional candidates exist for vaccine-directed immunotherapy. But what are some additional strategies that could be used to overcome the challenges associated with GBM? So the conventional mRNA vector is the most commonly used in clinical trials. But the main drawback is the limited antigen expression, which is proportional to the number of mRNA transcripts that are delivered. One way to overcome this limitation is by using self-amplifying mRNA vectors, which lead to increased production of antigens. And clinical trials are underway to evaluate these types of mRNA vectors. When looking for new antigens, most of the research is focused on indels and non-synonymous single nucleotide variants, or SNVs. However, many SNVs are actually patient-specific. Therefore, searching for additional sources of cancer neoantigens such as gene fusion, alternative splicing variants, and post-translational modifications may provide new targets for immunotherapy. The study that I presented revealed distinct neoantigens targets for vaccine against gliomas. Therefore, in addition to new sources of neoantigens that are potential solution to discrepancies, we could also personalize the mRNA vaccine based on a patient-specific tumor signature and deliver an mRNA vaccine that has several neoantigens that are unique to the patient's tumor. Lastly, given the extensive systemic and local immunosuppression in GBM, additional traditional adjuvants or also adding immune checkpoint inhibitors may be required in order to produce an effective vaccine against GBM. In conclusion, given the promising results of mRNA-based vaccines against pancreatic cancer and the recent advances in CAR T cells produced through mRNA technology, glioblastoma will also likely be treated with mRNA vaccines in the future. However, given the specific challenges associated with treating GBM, additional strategies such as finding other sources of neoantigens, personalizing vaccines using CAR T cells-based mRNAs, self-amplifying RNA vaccines, and adding adjuvants and immune checkpoint inhibitors might all be needed. And with that, I want to thank you for your attention.